everyone. Thank you for your attention. My name's Christian Jordan. I am getting my Ph.D. from UVM in 3 weeks in molecular biology. Thank you. After 4.5 years, and I'm going to talk to you about this fascinating field of science that's just emerging that explains why your DNA is not your destiny. So one of the things that led to the discovery of this field, which is called epigenetics, which just means above your genetics, is twins, identical twins specifically. So identical twins come when a single fertilized egg splits into two. So both of these people have literally the exact same DNA. But what happens when one twin, as it did in the case of these Spanish twins, gets diagnosed with cancer? Is the other twin supposed to just sit there and wait for the disease to inevitably come upon them as well? And the answer is no. It's not uncommon for one twin to get one dread disease and the other not. So the question is, how is it possible that these two people who are genetically identical, they're so similar, their bodies behave so differently? And so science has come to realize that your DNA isn't everything. These two mice, for example, these are clones. They're also genetically identical, but they look so different. And the reason they're so different is because the gene that is responsible for their fur coloring and the deposit of fat tissue is methylated in one mouse, and it's not methylated in the other. So what is this word methylated? So the word methyl group is just the word that the chemist came up with to describe a carbon atom with 3 hydrogen atoms attached to it. So these carbon atoms will attach to the DNA at particular genes and control them and suppress them or activate them. So it's sort of an on and off switch. So if you think about your genome, your DNA that you were born with, as a computer that can do everything that you'll ever need it to do, you can think of the epigenome, the collection of these carbon atoms attached to your DNA, as the software that controls it. So that's a really good analogy to think about this difference, and it makes sense because think about it. Every single cell in your body has the same DNA because it's your DNA. But they're all behaving differently, they all look different, and so the way in which the same genome can tell cells to turn into brain cells or heart cells or blood cells is because their DNA is being controlled by these methyl groups. Another fundamental function of the epigenome of these things is they serve as an interface between the environment and your DNA. Cells need to adapt to their environment in order to survive. That's a universal requirement of life here on Earth. And one way that this happens is through these methyl groups. And how do we know this is the case? So identical baby twins, if you look at the pattern of their methyl groups on their DNA, they're very similar to each other. But if you compare that to older twins, you'll see that their methylation pattern, which is the same thing as this epigenome, have accumulated differences as a result of exposure to the environment. Actually, here at McGill University, close to us, they found that rats that were nurtured versus not nurtured as pups behave very differently later in life. And so they're like, how do these rats remember what kind of care they received when they were babies? And part of the answer is this that I'm talking to you about. Finally, when you were conceived, you got one copy of your genome from your mom and another copy from your dad. So you have 2 copies of every single gene in every single cell. However, these 2 copies are not equivalent. For example, if you delete a small portion of chromosome 15, you will lose some genes and you will have disease. The interesting thing is that if the deletion is on your mom's copy, you're going to get one disease. If the deletion is in your dad's copy, you get a different disease. But it's the exact same sequence that's being deleted. So this is just a painting of a patient with Prader-Willi syndrome, and this is a patient with Angelman syndrome. And that's the region of chromosome 15 that's deleted in both cases. but because their epigenome is different, because the methyl tags are different on your dad's copy versus your mom's copy, the genes that are on in your mom's copy are different than the genes that are off in your that are on your dad's copy. So these methyl groups, this epigenome, is literally working as an on and off switch for your DNA. So we've sequenced the human genome about a decade ago, and it's not the end of the story. It's actually the beginning, because it doesn't tell us when these genes are being expressed, where they're being expressed, and it doesn't tell us any information about where these methyl tags are. So I hope I've convinced you that there's a whole new dimension of information in your genome, in the what we call the epigenome, that's important for disease, for development, for your environment. And I'm sure that if I gave anyone here in this group a piece of wood and a nail, you'd be able to get it through with a screwdriver if you really had to. But that's not ideal. And that's the way that we in the science field have been working with epigenetics. We don't have the best tools. So I was at Cold Spring Harbor about 10 years ago now, 9 years ago, and my boss told me, I want you to look at the epigenome of ovarian cancer cells and compare them to healthy cells. And after 3 months, I was like, I feel terrible, I've failed, I can't figure this out. And he said to me, well, you're the sixth person I've given this project, and you're the sixth person to have failed. I'm turning the project off. And I'm like, no, this is important, like, don't give up. And so this is where what drove the founding of Aquarius Life Science, which is a small molecular biology lab here in Burlington, Vermont, with the mission of developing a better test to be able to detect these methyl groups on DNA and see what kind of information it can provide about human health and how it can further medicine. Thank you.